RNA made easy. Hi guys, today's video is gonna be about the fundamentals of RNA, how it works, what it does, and more importantly, the structure of it, because we're gonna need to know about the structure of RNA to understand what is to come. But we will also take a brief look at its functionality or its main functionality. But first, hi everybody, I would like to welcome you to the channel, Biotech Lucas. Here we look at concepts and topics relating to biotechnology twice a week, Thursday and Sundays. And one thing to note before I start this video is that this is going to be building heavily upon my DNA Made Easy. I'll leave a link in the description, so if you haven't checked that out, or if you are very new to DNA, RNA, I highly recommend that one. I'm still going to try to make things as easy as possible so that you can follow along even if you haven't seen that video. Okay, but let's dive into it. And as usual, let's start with the definition. So, RNA. RNA is a ribonucleic acid, a nucleic acid present in all living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses RNA rather than DNA carries the genetic information, even though that is the function that DNA usually is responsible for. So this was actually quite a good definition because it already touched on one very central role of RNA, namely that it acts as a messenger. It takes the genetic information and carries it out of the cell nucleus so that the information can be used. Because if you remember, or if you haven't seen the video, DNA is the genetic code, the building instructions that the body needs in order to make you. However, DNA is confined to the nucleus of the cell, meaning that it cannot actually reach. You cannot take out the DNA from the nucleus in order to relate the information directly to the cells that are actually responsible for, or the parts of the cells that are actually responsible for protein production and making the building blocks to make you. And this is where RNA comes in. RNA is able to act as a messenger, and we will take a closer look at exactly how it does that in future videos, but we will begin to understand it today. One central principle that I like to remember to help me remember all of this is this right here. I'll make it red. DNA makes proteins. DNA makes RNA makes proteins, or DNA to RNA to proteins. That's just how I like to write it down so I remember it. Oh, never mind, that looks horrible. Sorry. Yeah, so you can write this down, and this is almost like a dogma in biology. This is not always the case, but very often, so it's good to remember. I also like to picture it as a library. So say that the DNA code is the library. You have a lot of information there, but you cannot just pick up the library and walk away with it. You cannot use the library for anything. Instead, you have to lend out the books. So you can think of the library as the DNA, the genome, if you will, made up of all of your genetic information, and then the books that you take out of it is small pieces of RNA, which is how you can actually extract the information out of the library. So let's jump into the structure. Again, this is going to look quite similar to my video on DNA, and that's the point, because RNA is quite similar in its structure to DNA. So I try to draw it in kind of the same way, but I'll try to make it understandable regardless. So let's go. The RNA is quite similar to DNA, as I already stated, but here already we see one major difference, namely it's a single helix instead of a double helix, which is the form that you might be more familiar. Usually we have this twisting staircase, basically with a line on both sides. This is probably a shape you're familiar with. But here we have a single helix instead of a double helix. Untwisting the helix and looking at it on a ladder model, you're going to see that RNA can bind to both RNA and DNA, which of course DNA can as well, but usually DNA is only bound to DNA, and then temporarily it binds to RNA. RNA is a bit more all over the place. Sometimes it binds to RNA, then it binds to DNA. We're going to have a closer look at that when we discuss DNA transcription and translation and protein synthesis. Those are coming up really soon. But for now, just know that RNA can bind to both RNA and DNA, and it does so regularly. We'll take a closer look at this as well later. There's one more important difference that you might have already noticed if you check the DNA video, but I'm getting ahead of myself. Then if we zoom in on a single RNA molecule, again it looks very similar to DNA. You have a sugar, you have a phosphate, and you have a nitrogenous base. Here's the first difference. In DNA, you have deoxyribose. Here you only have the ribose, and that's where the difference in name comes from. Ribose, RNA, deoxyribose, DNA. Here's the final difference, which are important to what we're talking about now. One of the nitrogenous bases are different. Can you figure out which it is? If you are familiar with DNA, this is probably new to you. Uracil. So instead of thymine as we had before, we now have uracil. So let's jump back to the stepladder model. Now we're gonna have, if we're gonna have RNA that binds to RNA, if we have an adenine or an A, instead of a thymine or T, we're actually gonna have a U. The cytosine binding to guanine is not going to change at all. That's going to be exactly the same. It's just that the T is going to be replaced by a U. And one way to remember this is just, I usually think of in what order these letters appear in the alphabet and think that the two middle ones go together and the two on the end goes together, because that works on both. Because C always go with G and A either goes with T, which is last in the alphabet when it comes to DNA, and U, which is last in the alphabet out of these four when it comes to RNA. So that's a good way, for me at least, to remember it. Then when it comes to DNA, if we have an A, an adenine, on the RNA, it's going to bind to a thymine on DNA. Because again, the base pairs are different. If we have a uracil on RNA, we're going to have an A adenine on DNA. So hopefully you're getting a hang of it. We can do a test, a short little test. I'm gonna write, that's gonna be a part of RNA, and this is gonna be a part of DNA. So what's the RNA gonna look like here? Try to think about this before I write it out. So an RNA, when it does complementary base pairing, as this process is called, the A is going to bind to U, correct? U is going to bind to then an A, correct? G, C, C, G. With me so far? Excellent. And again, now if it's a DNA, it's almost exactly the same. Here, C is going to bind to G, just as usual, G to C. T, which one is it going to be? It's going to be an A, right? And A is going to be a U. And the reason why we keep repeating this is because it's really important when it comes to understanding and reading the DNA code later on when we talk protein synthesis, because then you can actually understand how just these four different types of nitrogenous bases, which come in different order, translates to amino acids, which then create proteins. Okay, the final part of this is going to be the different types of RNA, and there are three main types of RNA, which are mRNA, which stands for messenger RNA, rRNA, which is ribosomal RNA, tRNA is transfer RNA, and these again are going to become more clear what these are once we move into the later videos. I keep referring to later videos, but it's better if we break them down, I think, video by video, so we can cover one concept per video so you don't hit an information overload. So just focus now on understanding what's the central role of the RNA. Remember, if DNA is the library, RNA is the book that we take out to extract information. How the structure is different, a single helix instead of a double helix, how it actually performs the complementary base pairing, A to U instead of T, and that on a molecular level, the sugar is different and one of the bases is different. U instead of T and ribose instead of deoxyribose. That should cover it. If you found any of this helpful, I really appreciate all the support you can give, whether that's a like or an additional question. And if you find this interesting, I highly recommend that you subscribe, because once we get through with all of the basic concepts, we can move into the more interesting and fascinating things. At least I think they are fascinating. How we can actually use all of this and how we are using all of this with the technology we have today and the technology we might have in the future. Yeah, have a great rest of the day. This is Biotech Lucas signing off. Bye!